Hello, this is Davina. Davina teaches biology. It's good to be back. I'd like you to subscribe to my channel so that you can be alerted whenever I make new videos. One of my subscribers requested flashcards for a particular topic. In a previous video, I had talked about how you should make flashcards for A-level biology. The key points: one definition or diagram or question per flashcard. You shouldn't try to put lots of different questions or definitions on one flashcard. It should be a quick flash, not reading paragraphs. So the topic that was requested was enzymes and transport in plants. So today I'm going to do flashcards in real time for enzymes topic. These flashcards would overlap for many different specifications. I have taught OCR, AQA, EDEXCEL, and even the Welsh exam board. So these flashcards would be suitable for more than one exam board. In terms of where to get the information from for your flashcards, the most reliable source is your textbook. I'm not using a book because I've been teaching this subject for many years, so I have the mark scheme phrases memorized already. But if you're learning A-level biology for the first time, or you're revising in year 12 or year 13, the most reliable source is your textbook because it will be accurate. Sometimes students are adverse to using their textbook and would rather use lots of different websites, but they're not always a trusted source of information, and you could be learning definitions that you think are great, but in actual fact, they're not really very accurate. So let's get started. Enzymes topic. My first flashcard, enzyme structure. Sounds really easy. Can you think of 3 bullet points that we can put on this flashcard? First bullet point for enzyme structure. Enzymes are proteins. Enzymes are tertiary proteins. Enzymes are globular proteins. And enzymes have a specific active site. So as I said to you before, I'm writing these flashcards in real time. So that is how long it would take you to write a flashcard for enzyme structure. Next flashcard, enzyme function. From GCSE, you might remember that enzymes are biological catalysts. That key fact is still correct at A-level biology. Biological catalyst means that enzymes speed up chemical reactions in living organisms. So on the front of my card is enzyme function, and on the back, biological catalysts. Next card, mechanism of enzyme action. You might notice that I'm not making up questions for each flashcard. You don't have to make up questions. Your flashcards just need to have key definitions, key facts. The exam will have questions, but you don't need to make up questions for your flashcard. I don't find that it's very helpful for students to do that. If anything, you spend more time trying to think of, well, what question could it be? So, mechanism of enzyme action. In GCSE, you learned lock and key hypothesis. That hypothesis is still correct now at A-level. However, we have developed upon that idea, and in A-level biology, we now focus on induced fit instead of lock and key. In the induced fit hypothesis or model, the substrate is not completely complementary to the enzyme's active site. However, Once the specific substrate does bind to the active site changes shape. The active site will mold around the substrate and it will put strain on the bonds. Another word for strain could be distort or stress the bonds. Because the bonds in the substrate are now under strain, it's much easier for those bonds to be broken. We say that the enzyme has lowered the activation energy. Bonds in the substrate are broken and the product will be formed. The key vocabulary that you learned in lock and key theory we can still use now: complementary and enzyme substrate complex. So I'm going to now write this flashcard for mechanism of enzyme action. Lock and key hypothesis is the older theory, whereas the more up-to-date theory is called induced fit. The key points for this card: initially, the substrate is not completely complementary to the active site. And I'm writing AS for short for active site and SUB for short for substrate. However, the substrate then binds to the active site, forming an enzyme substrate complex. The active site changes shape to fit the substrate more tightly. This then places strain on the bonds in your substrate. We can use the word strained or stressed or distorted. This means there is a lower activation energy. To remind you, activation energy is the energy that is required in order to start the reaction. But if the bonds in the substrate are already weakened or strained or distorted, then you need less energy to break those bonds. The products are formed once those bonds are broken, and the products leave the active site. This card has quite a few points, but if you write these points as short bullet points, you're more likely to remember them compared to writing paragraphs. I'm going to show you this card. Theory. Pause the video, of course, so that you can read each of the points that I have written down, and then add them to your own flashcard. The next card I'm doing will be extracellular enzyme. These are enzymes which act outside of cells. Extra meaning outside. All enzymes are made within cells because the enzymes will be made at the ribosomes. However, the enzyme's action can be outside of the cell. An example is amylase. If you remember, this enzyme is in your saliva. So the enzyme amylase is secreted outside of the cells in order for starch to be digested. So I'll also add an example to this card. Amylase. Next card, intracellular enzyme. These are enzymes which act inside of cells. So I hope this has given you a good start for how you can lay out your flashcards, how they should be short sometimes, maybe 4 or 5 bullet points at other times. I'm going to make another video to continue with the enzymes topic. In that video, I'll be writing flashcards for Q10, inhibitors, cofactors. So please watch my next video if you'd like to see the rest of the enzymes topic. And of course, like and subscribe so I can continue this channel and help you guys with biology. Bye.